Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, right, so we're in the process of discussing IP3 receptor mediated calcium signals. Okay, right, so we have now discussed that IP3 receptor mediated calcium signals can come in these two different types. We've seen the local IP3 receptor mediated calcium signals, which are those calcium puffs, which occur when you expose a cell to a low concentration of the calcium mobilizing agonist, such as acetylcholine. Okay, and we've then seen the global calcium signals, which are those calcium waves which propagate across the entire cell, okay, repeatedly, one after the other, okay, which result if you have a high concentration of the calcium mobilizing agonist in the extracellular fluid. Okay, right. What we now want to discuss is the actual effectors of calcium signal levels. Okay, so when calcium goes up in the cytoplasm, so what is the question? What does it actually do? What behavior in the cell does it change? Okay, what proteins respond to that intracellular calcium signal, basically? Okay, so we can't go through all of this. There is a ton of the things that respond to calcium signals. Okay, however, we're going to go through some of the highlights, and one of the ones that we really want to discuss is something called CamKII 2. And the reason we want to discuss CamKII 2 is because CamKII 2 responds differently, basically, to high-frequency calcium signals. Okay, so high-frequency calcium waves compared to low-frequency calcium waves. Okay, so that ability to decode different frequencies is what makes CamKII 2 really interesting. So we want to have a look at that. Now, before we can actually discuss CamKII 2, we need to actually discuss Cam itself. Now, Cam is short for calmodulin, so let's discuss this protein called calmodulin, and then we can move on to CamKII 2. Okay, so calmodulin is the next topic then up for discussion. Okay, right, so calmodulin then. Basically, when we're talking about effectors for calcium signaling, proteins that are going to respond and change their behavior in response to a calcium signal come in two flavors. There are the proteins, the effectors of calcium signaling, which actually have a binding site for calcium and which can bind calcium themselves and which will then change their behavior once they have calcium bound. Okay? On the other hand, there are targets which do not actually have a binding site for calcium. Okay, and instead, the way that calcium going up in the cytoplasm affects them is that calcium binds to some intermediate protein, which then changes its behavior and binds to this effector and changes the effector behavior. Okay, and calmodulin is one of these intermediates which can bind to calcium when calcium signals occur, okay, and can then bind to effector proteins, a whole host of effector proteins. We will see several in this video, okay, a whole host of effector proteins, and then change their behavior. Okay, so it's like this intermediate through which calcium affects loads of downstream targets. Okay, so calmodulin then has two states. There is the state where it doesn't have any calcium ions bound to it, which is called apocalmodulin. And for short, apocalmodulin is abbreviated down to ApoCAM, like so. So CAM is the abbreviation for calmodulin, and people do put the capital C and the capital M with the lowercase a in between them. Okay, and then the other state is the state where you have calcium bound, okay, which is called the calcium-calmodulin complex. Okay, so Ca2 plus CAM here. Okay, and these have very different structures, basically. So I'm going to draw out pictures for the apocalmodulin, and then I'll contrast the calcium-calmodulin complex to the apocalmodulin. Okay, so let's start off with apocalmodulin then. Okay, so both apocalmodulin and calcium-calmodulin complexes have two lobes. Okay, One lobe is called the N lobe, and the other is called the C lobe. So the N lobe is the one that's closest to the N terminus of the calmodulin protein, okay, and the C lobe is the one that's closest to the C terminus of the calmodulin protein, and it is just a single protein, so it's not a protein complex, it's a single protein, okay. Now, each one of these lobes, the N lobe and the C lobe, has two binding sites for calcium. Okay, and both of these binding sites are EF hands, so those domains that we have seen before where you have a helix-turn-helix motif with that turn having lots of glutamate and aspartate residues within it and therefore being negatively charged and coordinating the calcium arm within it. Okay, so all four of these calcium binding sites, okay, two in the N lobe and two in the C lobe, they are capable of coordinating calcium, and they are EF hands. So I'll put the little abbreviation for EF hands here, so EFH there. Okay, right. Now, the N lobe and the C lobe of the apocalmodulin are linked by a linker. Okay, so I'll show this linker now. So here is the linker linking together the N lobe with the C lobe. Okay, now in apocalmodulin, this linker between the N lobe and the C lobe is a linear stretch of the polypeptide, okay, which will change in the calcium-calmodulin complex. In the calcium-calmodulin complex, where we're going to have calcium ions bound to these four EF hand domains, that linker is going to go from being a linear polypeptide to being alpha helical in nature, and we'll see that in just a moment. The other thing to note is how in the apocalmodulin here, the lobes are kind of bent back towards one another. That's also going to change in the calcium-calmodulin complex. So here is our apocalmodulin then. At the moment, it doesn't have any calcium ions bound in these calcium binding sites, which are EF hands, okay? And it will be floating around in the cytoplasm, and at that 100 nanomolar calcium concentration, which is the resting calcium concentration, there is no calcium bound to any of these EF hand domains here. Okay, however, when calcium then goes up in the cytoplasm, when we get calcium signals, for instance those calcium signals that have been produced by IP3 receptors, okay, what will happen is calcium ions can now bind in these EF hand domains of the lobes of calmodulin. Okay, and this causes a conformational change in the calmodulin molecules. So let me show you now what the calmodulin is going to look like in these calcium-calmodulin complexes. So, firstly, the lobes stop being bent back towards one another as they are in the apocalmodulin. Okay, and they move more straight apart from one another, like so. In addition, that linker between the two lobes has changed from being a linear polypeptide to being alpha helical in nature. Okay, and this is the change that you've got when two calciums are bound in each of the EF hand domains on each of the lobes. So here are two calciums bound to these two calcium binding sites on this N lobe, and here are two calciums bound to these two calcium binding sites on the C lobe here. Okay, and this then now, this is a calcium-calmodulin complex. So when you get calcium signals occurring within the cytoplasm of cells, the apocalmodulin molecules start binding calcium ions, and they change from being apocalmodulin molecules to being calcium-calmodulin complexes. Now, this central alpha helix here of the calcium-calmodulin complex here, this is capable of a lot of flexibility. Okay, it's capable of winding around proteins. So let me just draw a picture here. So let's say that this is a portion of some protein here. Basically, the central helix of a calcium-calmodulin complex is capable of winding around proteins. So potentially we might have something like so. Here is this N lobe of the calcium-calmodulin complex. Then here is the central helix looping around the back. So I'm now showing you what you wouldn't be able to see because it would be behind the back of this portion of this protein. And then we'll have the C lobe maybe down here. Okay, so if I now colour in that protein in vivid purple, then you won't be able to see the central helix behind, and you'll only be able to see the bottom of the coelom peeking out from underneath there, like so. So basically, calcium-calmodulin complexes can wrap themselves around portions of proteins in this way that I've shown you here. Okay, using this incredibly flexible central helix that they have. Okay, right. And when these calcium-calmodulin complexes bind in this way to proteins like so, they can then change the behavior of the protein that they're bound to. Okay, and this is how they are this intermediate for calcium signals. Okay, they're a way that calcium signals can have a broad effect on many proteins that don't have calcium binding sites themselves. Okay, so the calcium works through calcium-calmodulin complexes. Okay, right. So now what we want to talk about, now we're armed with our understanding of calmodulin, okay, and how it can pass the calcium signal onto many effector proteins. Okay, let's now talk about CamKII 2. So firstly, let me tell you what CamKII actually stands for. So there is a large family of enzymes called the CamKInases, okay, written like this in short. Okay, and this in full is the calcium-calmodulin, okay, which I'll abbreviate down to CAM, dependent kinases. Okay, so CamKInases, really their full name is calcium-calmodulin dependent kinases. Okay, but everyone just calls them the CamKInase enzymes. Okay, now the CamKInases are actually a large family of enzymes. However, by far the most interesting one and the most studied one is this CamKII 2 enzyme. And this one has been found to be able to respond differently to different frequency calcium waves, which is what I want to discuss with you. Okay, right, so we are going to focus on CamKII 2 more than any of the other members. Okay, but it's worth just saying that all of these enzymes are serine-threonine kinases. They can phosphorylate serine and threonine residues within proteins and therefore change the behavior of a huge number of proteins. And they are important effectors of calcium signals. So when you get calcium signals in the cytoplasm of cells, you're going to get calcium-calmodulin complexes being formed. These will then activate calcium-calmodulin-dependent kinase enzymes, the CamKInase enzymes, and then the CamKInase enzymes will phosphorylate many other targets, and that will propagate the signal further. It will affect the behavior of a huge number of targets, and that's one of the means by which calcium signals can have a very broad effect on the cell. Okay, so we're now going to focus in though on this specific example of a CamKInase enzyme, which is the CamKInase 2 enzyme. Okay, right, so CamKInase 2 is a rather extraordinary protein. It assembles into massive great complexes of 12 individual CamKInase 2 proteins. Okay, so it assembles into dodecamers, basically, 12-membered structures. Okay, however, before I show you the dodecamer of CamKInase 2, I want to firstly just show you a CamKInase 2 monomer. Then I'll talk about how it assembles into the dodecamer, and then I want to talk about how we can activate the CamKInase 2 enzymes within the dodecamer, and then we'll talk about how the CamKInase 2 has this memory property, which then allows it to start responding differently to different frequencies of calcium waves. Okay, right. So let's start then by discussing the structure of a CamKInase 2 individual proteins, one monomer of CamKInase 2. Okay, right. So we'll start off from the end of the protein rather than the beginning of the protein. So we'll start off with the C-terminal portion of the protein here. Okay, and one of the really important domains of the C-terminal of the protein is something called the hub domain, and that's what I'm trying to show here. Okay, and I want to try and get it looking a bit more 3D, so I'll just add that portion in here. So our colour is in, that will always make it look better. Okay, so here in blue, this is the domain of CamKInase 2 called the hub domain, and this is going to be really important in getting the CamKII 2s to assemble into this dodecamer, basically. So this is going to be important in oligomerizing the CamKII 2 enzymes together. So this is a hub domain. Okay, and this is at the C-terminal end of the polypeptide. So if you like, there's the C-terminus of the polypeptide. Okay, then going towards the N-terminus of the polypeptide then, off the hub domain, what we then have is a domain called the autoinhibitory domain, okay, which is represented by this rectangle here, which I will colour in turquoise. Okay, and this is going to be important in inhibiting the kinase domain, which I'm about to show you. Okay, so this is the autoinhibitory domain of this individual CamKII 2 monomer. Okay, and then after the autoinhibitory domain of our CamKII 2, the final important domain that I'm going to show you is the catalytic domain, or the kinase domain of the CamKII 2 monomer here. Okay, so this is the domain that's actually capable of phosphorylating serine and threonine residues within proteins. So this is the actual kinase, basically. This is the one that can add phosphate groups onto serine and threonine residues within proteins. Okay, so I'll highlight this up in red here. Okay, right. So in red there, that is the catalytic, or we could call it the kinase domain, so catalytic / kinase domain, and it's been represented in the typical way of showing an enzyme where we've got the active site represented here. Okay, and then we've got the N-terminus of the CamKII 2 monomer. So this is how I'm going to represent an individual CamKII 2 monomer. It's an individual protein. Here's the N-terminal. Here is an important domain, the domain that's actually involved in catalyzing the addition of the addition of phosphate groups onto serine and threonine residues. Then we've got the autoinhibitory domain, which is going to be important in inhibiting the catalytic domain. Okay, basically this is going to insert into the active site and block the active site from actually phosphorylating any serine and threonine residues. And then we've got the hub domain here, which is important in oligomerizing the CamKII 2 proteins together. Okay, so making them form this dodecamer. Okay, right. So now what I want to do is actually show you the CamKII 2 dodecamer. Okay, how do these CamKII 2 monomers assemble into a dodecamer? And I want to show you at the moment the inactive dodecamer. Okay, so when I've shown out this monomer, I have shown you it with the autoinhibitory domain not actually in the catalytic domain, so this would actually be an active monomer, okay? I did that just to make it look simple, okay? But in reality, without a calcium signal and without calcium-calmodulin complexes, the CamKII 2 will be in the inactive state. So when I now go over to showing it as a dodecamer, I'm going to show the CamKII 2s in the inactive state. Okay, right. So firstly then, how does it actually assemble into this dodecamer? Well, what happens is these hub domains, I've tried to show it as basically a slice of pie here. Okay, and what's going to happen is this is going to get together with some other hub domains and they're going to form a ring. Now, you do not form a ring of 12 hub domains. Instead, you form a ring of 6 hub domains. So let me show you the ring of 6 hub domains here. Okay, so this is now a ring of 6 hub domains here. Okay, all put together. So I'll split it up into 6 separate hub domains, like so. So you have brought together then 6 CamKII 2 proteins. You have put all of their hub domains together to make this pie. Okay, and you've now got 6 CamKII 2 enzymes all put together. Okay, and I'm not going to draw every single one of their other portions coming off, but every single one of them will also have this autoinhibitory domain with the catalytic domain coming off the side. Okay, I'm not going to draw it for every single one, but I will draw it for one in a moment. Okay, however, we've got a more pressing problem at the moment. I've told you that these things form dodecamers. At the moment, we've got a 6-member dodecamer. Okay, how are we going to turn this into a 12-member dodecamer? Well, basically what you're going to do is you're going to assemble one of these 6-membered oligomers, then you'll do it again, you'll get another 6-membered oligomer, then you'll stack them one on top of each other, okay, upside down basically. So you're going to stick another one of these oriented in the opposite way, okay, underneath this one here, okay. So basically this one is up one way and the other one is upside down relative to that one. Okay, so let me colour in this 6-membered oligomer of hub domains also in blue down here, and that will overall now give us a dodecamer where we've got 12 CamKII 2 enzymes all stuck together in a great big complex. Okay, so once again I'll stress the point that this one is facing one direction and the other one's upside down relative to it, so you've effectively turned this one upside down to stick it on there basically. So take this one, spin it upside down and then stick it on there, and then you've got your dodecamer basically. Okay, right, so what I now want to do is show the other portions of the CamKII 2 monomers for a few of these hub domains that we've got. Okay, so basically all of the CamKII 2 monomers in this CamKII 2 dodecamer are going to initially be in the inactive state, okay, where they have the autoinhibitory domain bound within their active site, and therefore their active site is not actually active. Okay. However, there are two different ways that this inhibited CamKII 2 monomer can actually orient itself basically. So let me show you this. So I'm going to firstly show you one way in which an inhibited CamKII 2 monomer can exist. So here now, this is the autoinhibitory domain here. Okay, and then we'll have the catalytic domain attached to the inhibitory domain here, like so, with that inhibitory domain basically sat in the active site of the catalytic domain. So here in red, this is the active site of the, this is the catalytic domain rather of our CamKII 2 monomer here, okay, and then we've got the autoinhibitory domain here, which is inhibiting the active site of that catalytic domain. Okay, so this is one conformation then that this inactive CamKII 2 monomer in this dodecamer can exist in. Okay, and this is what we might call the closed conformation. Okay, because when the CamKII 2 is in this conformation, it is inactive. Okay, it's inactive because the catalytic domain is being inhibited by this autoinhibitory domain, but it's not actually even activatable. Okay, calcium-calmodulin complexes are not even actually capable of activating the CamKII 2 monomer when it's in this conformation. Okay, in order to actually be activated by calcium-calmodulin complexes, it needs to move into a more open conformation where calcium-calmodulin complexes can actually gain access to it. So now let me show you a more open conformation. So a more open conformation might be like so. Okay, here's the inhibitory domain no longer pressed against the hub domain but more open now. Okay, and it's still an inactive conformation because the catalytic domain of the CamKII 2 is now still going to be attached to that autoinhibitory domain and therefore is going to be inhibited by the autoinhibitory domain. So let me just colour that in. There's the autoinhibitory domain there in turquoise. Okay, and here is our CamKII 2 catalytic domain in red. Okay, so this we might call the open conformation then of our calcium-calmodulin dependent kinase 2 monomer here. Okay, so basically both of these states are inactive. Okay, both of the catalytic domains here are bound to the autoinhibitory domain and therefore are not capable of catalyzing the phosphorylation of any serine or threonine residues. Okay, however, they are in different states, these two CamKII 2 monomers in the dodecamer. This one is very closely tucked up to the hub domain. Okay, and that means that it's actually not available to bind to calcium-calmodulin complexes. So calcium-calmodulin complexes can't gain access to this autoinhibitory domain to change its conformation and stop it from inhibiting the catalytic domain. Okay, whereas this other CamKII 2 monomer here, this is in this more open access conformation where the autoinhibitory domain is more available now for calcium-calmodulin complexes to bind to it. Okay, so basically in the calcium-calmodulin dependent kinase dodecamer here, some of these calcium-calmodulin dependent kinase 2 monomers will be in the closed state inactive state, and some will be in the open state inactive state. Okay, so some of them here will be in a state where they can actually be activated by calcium-calmodulin complexes if calcium-calmodulin complexes are formed in the cytoplasm of the cell, whereas others are in a state where they cannot be activated by calcium-calmodulin complexes even if calcium-calmodulin complexes are formed within the cell. Okay, now what that actually means is that when calcium-calmodulin complexes are formed within the cell because there has been a calcium signal, not all of the CamKII 2 monomers in a dodecamer are actually going to be activated. Only the ones that are in the open state are actually going to get the calcium-calmodulin complexes binding to them and resulting in them changing into the active conformation. Okay, and that will become really important. Okay, so we'll call it there for this video, and in the next video what we'll talk about is how calcium-calmodulin complexes can actually activate these CamKII 2 enzymes. Okay, and then we'll talk about the memory property of the CamKII 2 dodecamers, and then we'll move on to how it can actually decode frequency of the calcium waves in a cell.